Hello, everyone. I'm Jeremy Wilkinson, PacBio's market lead for microbiome and metagenomics, and I want to welcome everyone to the PacBio webinar series with today's topic focused on the recent advances by PacBio in shotgun metagenomics that are leading to improved results, capacity, and cost effectiveness in microbial community profiling and MAG assembly. I'm joined today by two great researchers who will present in the following order: Dr. Xiaowen Feng, a postdoctoral fellow at the Dana-Farber Cancer Institute and Harvard Medical School, and Dr. Daniel Portick, a senior bioinformatics scientist here at PacBio. We have a lot of great material to cover today, and the presentations portion of the webinar will be followed by a question and answer session. And you are welcome to submit your questions at any point during the webinar by typing them into the area provided on your attendee control panel, and we'll get to them at the end. We've also uploaded a number of complementary pieces of literature to your control panel, so please feel free to download those and take them with you. We'll be recording this webinar and making it available for download in the next few days. So please keep an eye out for a follow-up email with a link to the recording. So with that, let's get started with a brief overview of PacBio's technology capabilities. So as I assume you well know, PacBio is providing solutions for complete and accurate long-read sequencing, most recently with the Sequel IIe system. While we've done so for over 10 years now, we've seen a particular enthusiasm and adoption through the development of HiFi reads. These are sequence reads that are uniquely both long, around 15 to 20 kb currently, and highly accurate, so 99.9%. And they've been demonstrated to have a powerful utility in many areas of genomics, whether it be genome assembly, detecting all variant types, targeted sequencing, long-read RNA sequencing or iso-seq, metagenomics or epigenetics, so single molecule methylation calling. HiFi reads provide a new lens to interrogate living systems and thereby allowing many discoveries that are not possible with other technologies. So HiFi reads uniquely combine extraordinary accuracy with long read lengths, and these qualities, along with low GC bias and single molecule resolution, really make PacBio Smart Sequencing the best-in-class technology for a wide range of applications, including in microbiology. HiFi reads are typically 15 to 20,000 bases in length, which is about 100 times longer than your typical short reads. The raw polymerase reads in PacBio sequencing have become so long now, certainly well over 100 kb in length, which allows for building consensus from multiple passes on a SmartBell insert template, which leads to that 99.9% inaccuracy of the HiFi reads. The sequencing errors here, as indicated in these tiny red dots, they're random, and therefore they'll be washed out by increasing those number of passes around that SmartBell. And here on the right compares the read accuracy distributions of the three major sequencing technologies using the most recently publicly available data sets for each one. And you can see that PacBio HiFi reads here in green are very close to Illumina reads in yellow, and they're extremely accurate, so 99.9%. This is in contrast to the other long read sequencing technologies here in purple that are much less accurate. Secondly, the HiFi read distribution is very narrow, which makes the bioinformatics much easier and much faster because the algorithms can assume this very narrow range of high accuracies. This is also very different compared to the other long read sequencing method, which you can see it's a very wide distribution as well as a long tail. So the PacBio Sequel IIe system is a powerful tool for understanding microbial genomics, and these are through a variety of different approaches and applications, such as whole genome sequencing of isolates of single organisms to get reference quality genomes and most of the time single contig genomes, metagenome or microbial community sequencing, and targeted sequencing assays such as our HiFi viral for COVID surveillance. But today's webinar, we're going to focus in on this category, so metagenomics and specifically shotgun metagenomics. So there are really three ways that you can use PacBio Smart Sequencing to understand microbial communities. These are through full-length 16S sequencing or really any other full-length gene sequencing. This can be fungal ITS or eukaryotic 18S, for example. And this really reveals the true diversity of your samples with better taxonomic resolution and can generate more testable hypotheses. HiFi shotgun metagenome profiling provides more and richer functional information without error correction or assembly and can reveal the true positives and taxonomic profiles with high precision. And then for metagenome assembly, HiFi reads provide far more high-quality metagenome-assembled genomes, or MAGs, than other technologies, and more complete circular and single contig MAGs, even at lower coverages. And all of these are made possible because PacBio HiFi combines both long reads and high accuracy. And you don't have to just take my word for it. Here are some select quotes from recent customer publications. So full-length 16S sequence data has the potential to enable accurate classification of individual organisms at very high taxonomic resolution. Long-read approaches provide valuable additional information, including greater taxonomic resolution with long-read amplicon sequencing and improved functional profiling and bacterial genome recovery from long-read metagenomics. And HiFi reads allow us to generate a nearly complete picture of the metagenome, not just a fragmented assembly. And like complete genomics, which is already being applied to rare disease diagnostics, complete metagenomics may soon make its way into medicine and many other disciplines. So there are several recent publications which all describe the same result. PacBio HiFi sequencing represents a new gold standard in shotgun metagenomics. The long and accurate HiFi reads provide substantially more resolution and a higher number of complete MAGs compared to other technologies, whether this is applied to the gut microbiome or other microbial community samples. Bioinformatic methods have also been evaluated and benchmarked, as well as applied to characterizing even low abundance species. And you'll actually hear about some of these studies in today's webinar. And so through improved performance and optimizing our pipelines, which you're going to hear about some in today's webinar, is leading towards higher capacity and throughput, which leads to more cost-effective projects while providing superior results. So here's just some example estimated costs for the three main applications. So for full-length 16S, or like I said, any other gene of choice, it's about $8 per sample. For shotgun metagenome profiling, we used to recommend up to 8 multiplex samples per SMRT Cell, but due to a recent downsampling experiment that Dan has performed, which he'll mention a little bit today, we now can recommend up to 48 samples multiplex on a single SMRT Cell, which is less than $100 a sample. I do want to mention that this recommendation is for standard community taxonomic and functional profiling using our optimized PacBio GitHub pipelines that you'll hear about today. And if interested in more in-depth functional profiling analysis, so really wanting to get every single gene annotated for functional annotation, then you may want to target less multiplex samples. So probably something around the 8 or less than that range. For a shotgun metagenome assembly, this one's really dependent on how diverse your community is and the research question in mind. It could be up to 4 samples multiplex on a SMRT Cell for $350 a sample, or you may want to get more high quality MAGs, more single contig MAGs. If that's the case, then you may want to do just 1 sample per SMRT Cell, or even a single sample across 2, 3, or 4 SMRT Cells. So that one really depends on the research question. So we have lots of resources available online, so best practice guides, plus protocols for both 16S and shotgun metagenomics, and also we have resources available again on that side panel that I mentioned. So with that, thank you for your time, and now I'll turn it over to our first speaker, Xiao Wen. Thank you.
Hello, everyone. Today I'll be talking about metagenome assembly with HiFi reads. I'll briefly introduce a new assembler based on our lab's previous works and share the evaluations and other findings in a couple of empirical datasets. The preprint of this manuscript is available on arXiv, as you can see the accession number here. The assemblies in a revised version of the manuscript are on Zenodo. Some numbers will be different, but what we said in the preprint remains mostly unchanged in the revised version. Assembling metagenome samples is an important way to learn about real-world microbiome community. The metagenome-assembled genomes, or MAGs, however, are often very fragmented due to the short read length in the second-generation sequencing and also the sample's complexity. Well, for human gut samples, the public databases have accumulated more than 200,000 MAGs. Around only 90-something were reported as single complete conics. Half of those are also assembled with error-prone long reads instead of the second-generation sequencing. To recover continuous and complete conics, it usually takes more than some manual intervention and some pipelines. The yield per sample was also limited. Well, with the exception of the Metaphy paper, whose work first described and assembled a HiFi metagenome library, which yielded 37 complete circular conics in 1 sample and 1 shot. So we extended a previous work from our lab, which is the HiFi assembler, to fully exploit the low error rates in HiFi libraries, but enter a metagenome setting. Metagenome assembly with HiFi reads or just metagenome assembly in general has several challenges. First, we do not know the total number of haplotypes, and these haplotypes also receive uneven coverages based on their concentration in the real-world sample. And handling read containment is also very tricky in this setting, which is complicated by the other two issues. HiFi Meta uses the same overlapping error correction workflow as HiFi with an optional read selection module for highly duplicated libraries. So far, this module is only useful in mock datasets, as we shall see later. Very high coverages in the real datasets are not almost not enough. And in this workflow, after the overlapping error correction, the assembly graph cleaning module is rewritten to manage the metagenome challenges. HiFi Meta is open source on GitHub. It can be compiled with Metagenomic or installed from Bioconda. First, we evaluated HiFi Meta in two mock datasets. In this category, Metafly, HiCanoe, and HiFi Meta are largely comparable. So in the first mock sample is the ATCC MSA 1003. 13 out of the 15 prevalent species down to the concentration of roughly 0.2% were recovered as complete circular conics. One of them was recovered as a almost complete linear conic, and the other one was recovered in two conics, also almost complete. The other 5 samples, their coverage was too low to assemble based on alignments against the reference assemblies. The second mock sample is the Zymobiomix D631. The prevalent species are assembled reasonably well. The main challenge of this community is that it has 5 very closely related E. coli strains and also a salmonella. However, this challenge is too difficult. Although we can get conics of around maybe 1 megabases long, it was hard to assemble all the E. coli genomes in full. So HiCanoe was slightly better than HiFi in this category. HiCanoe assembled 2 of the E. coli strains into complete circular conics. HiFi Meta got 1 of them and got the other one into 2 linear conics. Metafly was able to recover the B766 as 2 conics and missed the other one. But overall, the 3 assemblers are comparable in the mock datasets. Moving on to the real datasets, we found that because the lack of reference assemblies, we have to resort to the binning and evaluation procedure similar to previous short read-based studies. Briefly, we estimate the conic coverages using Minimap2 and MetaBat2's module, then bin the conics with MetaBat2 as the default the workflow. And we noticed that after binning MetaBat2 or other binners designed for short read assemblies, it would struggle with HiFi assemblies, probably due to the assemblers were being able to face very closely related haplotypes. Sometimes the binners will bin a long circular conic with other shorter conics, or occasionally even get 2 circles together. After some analysis, we decided to trust the assembler and split these bins in a post-processing step. We let the circular conics to form bins on themselves and left the other conics as a splitted bin without further treatments. After this step, we get the final binning result of the assemblies. For evaluation, we use CheckM following the conventional definition of the quality buckets. We define near complete mags as the mags that have more than 90% of completeness and less than 5% of contamination. 70% completeness and 10% contamination marks the high quality bucket, and there's a little bucket marking the medium quality, and the rest of the mags are considered failing the quality check. For real datasets, so far there are about 9 public HiFi libraries with sufficient coverage for assembly. We are going to cover 8 of them from here and in the manuscript. I want to mention that there was a dataset derived from seawater, which looked very interesting in the public domain, but not public domain, in the public repository. But unfortunately, the coverage was way too low to assemble. The libraries colored in orange here are sequenced on polled samples. Each of them actually presents 4 human gut materials. They also come with the host diet information. 2 of the libraries, as we labeled here, are from hosts with vegan diets, and the other 2 are from omnivore individuals. This labeling offers an interesting result, as we shall see later. The libraries colored in blue here are based on single samples. Notably here, SHIP A and SHIP B, they represent the same biosample, while SHIP B was targeting a much higher coverage, and SHIP B was sequenced solely on SQL2, while SHIP A was a combination of SQL1 and SQL2 libraries. The ground truth of these samples are unknown. Since HiFi reads are usually much longer than full-length 16S RNAs, we decided to identify the 16S genes with BARNAP and predicted the taxonomies with RDP classifier and the SELVA database. As shown in the lower part of the tables, the complexity of the polled libraries are similar to the single sample libraries, but obviously they have less overall coverage in terms of per sample coverage. This actually resulted in tangled assemblies. We assembled each sample with HiFi Meta, HiCanoe, and Metaphy. In these assemblies, the most interesting category is the long circular conics, as we would expect them to be able to directly serve as complete and closed genomes without any further post-processing. Indeed, most of these long circular conics they qualify as near complete MAGs based on ChIP-M and RNA evidences. They're plotted as green bars here. Overall, HiFi Meta provided better results. SHIP-A and Chicken assembled particularly well here. We recovered 134 and 62 near complete circular conics from them, respectively. The sludge sample and the human samples, on the other hand, reported less circular MAGs, maybe due to lower coverages. Binning was hard for the assemblies from all 3 assemblers. They often get bins obviously containing conics from multiple similar haplotypes. The 3 quality buckets for bin MAGs are plotted here in grayish blue. Looking closer to HiFi Meta's long circular conics, first, most of them are near complete in terms of ChIP-M. And in terms of RNA completeness, they almost all of them have all 3 types, along with more than 18 copies of tRNAs. And secondly, we found little duplication among these long circular conics. For pairwise comparison between all long circular conics recovered in the empirical samples, all pairs except 4 have at least 1% sequence divergence. The content of our conics are also highly consistent with the other 2 assemblers' report. For example, HiCanoe assembled 73 long circular conics in SHIP A. We recovered 61 of them as long circular conics with very low sequence divergence. Another 10 were recovered in long linear conics with similarly low sequence divergence. As mentioned before, the SHIP B sequences the same biosample of SHIP A, but with much higher coverage. Empirically, it seems that while having 50 gigabases, SHIP A still has not exhausted the sample's complexity. We recovered 132 more circular conics in SHIP B and saw a similar gain in the near complete MAGs recovery. Although this is a diminishing gain because SHIP B has 4-fold more the library size, but we are only recovering 2x more complete circular conics. Based on 16S RNA hints though, SHIP B might also not have exhausted the sample's complexity, as we still see a lot of NR-represented genera here. Although I'm not sure what's the upper limit and if this evidence is actually that reliable. And the other showcase is that recently there's a single sample library featuring a human gut sample became available in the public repo. Compared to the polled human sample libraries, this single sample gets roughly 5-fold more bases. I don't have conclusions from this assembly graph visually, but I think it's good to see that the large tangle that we see in the pooled sample does not persist into the single sample with higher coverage. On the other hand, this single sample assembly seems to imply that the human gut microbiome is less diverse than the sheep's and the chickens. I'm not sure if this is actually true. Just for reference, the sheep has more than 100 of long circular conics and the chicken has around 62, as I remember, and the human only gets around 36 here. And for detailed results on the effect of sequencing depth and tips, please stay tuned for Daniel's talk. The human library offers a very unique opportunity to compare microbiomes communities of different diet groups. We initially tried to evaluate using alignments, but did not find a sufficiently continuous reference assembly set. And the alignments between the de novo assemblies were complicated to analyze. So instead, we tried pulling all the 4 libraries and co-assemble. Effectively, this co-assembly's input has 16 biosamples, and HiFi meta does not make use of the reads origins. The total number of circular conics reported in this co-assembly were less than simply adding all 4 standalone assemblies, implying either collapsed haplotypes or there are newly unresolved regions in the co-assembly. Interestingly, however, most of the long conics in the co-assembly are either omnivore specific or vegan specific. This is also true for slightly shorter conics, like down to 500 kilobases. I forgot if this is true for all conics length ranges, but this is the trend. And for long conics, they show little to no in-group preference, taking reads from other sources. You can see here the human O1 seems to have a slight advantage over human O2, and similarly for human V1 over human V2. But this was probably arbitrary. We speculated the cause to be the read length or read quality differences between the libraries, but we did not investigate. Unfortunately, this is hard to replicate in short read libraries or assemblies, as short read assemblies usually use deep graph assembler, and that process will discard the read origin information. The 16S RNA-based taxonomy was confident at genus level, but it was hard to go lower. So we decided to use phylogenetic tree reconstruction to show if the long circular conics and genome bins are differentiable on maybe species level. This inference is done with GTDBTK. The MAGs are placed without obvious clusters, suggesting that the sample specificity we saw in the sample-specific conics is more subtle than what the phylogenetic inference could figure out. So probably at species level or even lower. There are 19 genera that have at least 3 MAGs here, and 16 of them have both omnivore-specific MAGs and vegan-specific ones. As a negative control, we tried co-assembling sheep A and chicken. Although these 2 samples are both speckle materials, we would expect to see species or lower level differences because the organism is pretty distinct. And indeed, in this time in the phylogenetic tree, the sample-specific MAGs form clusters on the tree. You can see the red labels are representing sheep A conics, and they tend to cluster together. And the blue labels are the chicken conics. They also tend to cluster on their own. Based on these observations, we would like to say that the differentiation between diet groups was real and possibly below species level, although it is hard to pinpoint where exactly the differences are. And from here, I would like to share some bits in development. As you can see in the assembly graph from this large sample here, we have some long circular conics, but we also have these kind of subgraphs where we have localized complex topology, but we can still somewhat see that there's, for example, here we have like 3 things. And we would like to speculate them to be haplotypes that's almost separated from others, but are still too complex to to be directly phased into a clean contig. It'll be interesting to either look closer at these so-called haplotypes or try to improve the assemblies of them from here. However, the graph itself is difficult to directly make use of. And also because of the similarities between the little conics in the tangles, binning based on the sequences and coverages will be very hard. The contig coverage information isn't very useful. Therefore, we propose a heuristic, topology-based heuristic to find the elementary circuits that might look like a MAG, remove the duplications based on sequence divergence, and then report the remaining paths separately from the primary assembly for the end user to have a closer look. Conics used by the pathfinding are colored in red here in this plot. This will introduce errors, but we think it'll be better than doing nothing. The user can choose to further curate the paths using CheckM or RNA evidences if they're interested in these haplotypes. We are still tuning the implementation, so the following results are reported with the help of CheckM and also MeshJSONs. The MeshJSONs is closely related with ANI, which is further closely related with the whole genome divergence. So I'll be just saying sequence divergence from now. In this large sample, the primary assembly reports 19 long circular conics, and CheckM thinks 15 of them are near complete. With the pathfinding heuristic and deduplication at 3% sequence divergence threshold, we can get another 17 near complete circular MAGs, colored in blue in this plot. We did a K-mer-based assembly completeness check. This plot is based on CAT, the K-mer analyzer. For each column in this plot, it accumulates K-mer that have a certain count in the HiFi reads, which is marked on the x-axis. And in the column, the set is further parted based on the K-mer counts in the conics. The y-axis marks the ratio of each partition of the columns. The color legends are here. So from the left to right, this graph roughly reads as the following. For K-mers that are rare in the reads, we don't see them often in the conics, which is normal because these K-mers are likely to be either sequencing errors or just reads from haplotypes that have very low coverage. And for K-mers that are prevalent in the reads, they're also likely to appear in the conics, as we see the area of color orange here. This is good because we are probably assembling those genomes with okay coverages. And also notice the absence of green or red areas in this range, which implies that we don't have a lot of duplications. And the K-mers that are very prevalent in the reads, however, we again don't see them in the long circular conics, which is not good because we are missing out the very prevalent haplotypes probably. Then we did the pathfinding rescue and deduplicated at 1% sequence divergence threshold. We combined these rescued paths with the real long circular conics and did the evaluation again. The blue areas towards the right end of the block shrink significantly, implying we are getting a more complete conic set. However, the green and the red area expands, suggesting that we have duplicated MAGs. So again, we lifted the deduplication threshold to 3% sequence divergence and try it again. It looks better. We have the orange area covering most of the K-mer frequency range, and we don't have a lot of green and red on the plot. For the blue peak towards the rightmost area, we plotted the K-mers and found that they are probably coming from RNA operons or some other mysterious genes that have no hits in the RNT database. I'm not sure what are those. And as a comparison, here's the assembly graph of the sheep A, which we think is a reasonably desirable outcome of metagenome assembly, and the K-mer plot of the sheep A assembly, the sheep A's long circular conics. As you can see in the blue area, we are still missing some of the haplotypes. I guess some of them will be recovered in the sheep B assembly. So to summarize, we present HiFi as a meta for HiFi genome assemblies. The goal is to recover closed and complete MAGs without manual intervention or any prior knowledge of the microbiome community. And similar to the HiFi upstream, it is a standalone executable. Evaluated in 2 mock datasets, HiFi is comparable to Metaphy and HiCanoe at the baseline. And in real datasets, depending on the sample's complexity and library size, we were able to recover tens to hundreds of circular MAGs in one shot. The results outperformed the current state-of-the-art assemblers, although binning in general still remains a problem for HiFi assemblies. And we saw some promising results in the post-assembly processing, although this part it still needs some work. I would like to thank my supervisor for conceiving and overseeing this project, and my coworker Hao Yu at the FCI, who's the author of HiFi. And thank you, Daniel, for the in-house evaluations and granting us access to the Zymo mock dataset for early development. And thank you, Wei and Picard, for the chicken and the SheepBee dataset data access, respectively. And finally, thank you. That's all.
Great work, Xiaowen. And thank you for all you've done. Next up is Dan Portick, and he's going to talk about precision species profiling and metagenome assembly with HiFi reads. Take it away, Dan.
Great, let me just go ahead and get set up here. Okay. Great, well, thanks for the introduction, Jeremy, and thanks everyone for being here today. So I'm Dan Portick. I'm a bioinformatics scientist here at PacBio, and I've done a lot of development for shotgun metagenomics and developed a lot of the pipelines and infrastructure we've been using to evaluate and do research on this topic. So in the talk today, I want to focus on 2 main topics. First is read-based taxonomic profiling, and second is metagenome assembly and evaluation. And for both topics, I want to provide you with some key pieces of information. So first, I want to give just a very simple overview of the analysis, what it is and what we hope to get from it. I'll then go into some detail about the pipelines we have available, as well as talk about some recent improvements I've made to them. Next, I'll show some example results using HiFi data. And then finally, I'll talk a little bit about what effect sequencing depth has on each type of analysis. So when we increase sequencing depth or decrease the sequencing depth. So we're going to start out with read-based profiling. So the goal of this analysis is to identify the species present in a sample and estimate their relative abundances. So in a given microbiome sample, we extract DNA, fragment it, sequence it. We then take this read set and attempt to match it to some type of a reference database. And ideally, that would allow us to classify the reads into different species or groups. And for each species, we know how many reads have been assigned, and this allows us to get relative abundance estimates. So there are a lot of methods that are available for this type of analysis, but nearly all of them have been developed to work with short reads, and only a handful have been explicitly designed to work with long read datasets. And so kind of an open question for the last couple of years has been, what is the best method to use for long read datasets, and in particular, HiFi data? And so I recently attempted to address this by performing a comprehensive benchmarking study. And this is a collaboration with Titus Brown and Tessa Pierce-Ward at UC Davis. And we evaluated a lot of different taxonomic profiling methods, and we used long read mock community datasets to do this. We found a lot of methods did quite poorly, but there were a couple that performed pretty well. And those top performing methods shared a couple of characteristics. So first, they used full nucleotide or protein alignments, and this is in contrast to using something like k-mers, which, although being faster, tends to produce more inaccurate results. They also used a last common ancestor algorithm. So rather than attempting to assign each read to the species level, in cases of ambiguity, they could assign reads to the genus level, family level, or higher. And this was really good at preventing misassignments. And finally, they used some type of minimum threshold filtering to sort through all the hits. So they didn't report all the hits, and this threshold filtering had a very positive impact on precision. So I just want to highlight our top 2 methods that we identified as sort of the leaders in their class. And so one of these is BugSeq, and the other is a PacBio pipeline that we have available. So BugSeq is a cloud-based platform. It uses nucleotide alignments. So you upload your data and download a report. It's fast, easy to use, and highly accurate. And the PacBio pipeline uses Diamond and Megan to do alignments to proteins. And this is the pipeline that I'm going to talk about today. And so let's go into a little more detail about what's happening with it. So first, I want to just point out that all of our pipelines are publicly available on GitHub. Under Pacific Biosciences, we have a repo called PB Metagenomics Tools, so you can find all of our workflows and documentation there. So to do taxonomic profiling using this method, it actually requires running two workflows. So the first one, the inputs are a set of HiFi reads and a protein database. And so the obvious choice is the NCBI non-redundant protein database, but it is quite large in size, so you could always use a subset of this, but it does require using proteins. And in the first step, we're performing a translation alignment of the reads to the proteins using Diamond. And we're using long read settings for this. And that is essentially guaranteeing that we're reporting hits along the full length of the read rather than having a lot of hits to one particular region. And so it's better at representing the full length of our reads. After alignment, we then convert those into a format readable by Megan. And along the way, Megan is doing some of its own internal processing. And so the output from this first workflow is a filtered version of this Megan file, as well as an unfiltered version of this Megan file. And I'll talk more about this filtering step in just a moment. And with these Megan files, you can then use the second workflow, where we're basically using Megan to interpret and summarize all of the alignment information. And then we're outputting taxonomic annotations, which is what we are after. But in addition to that, we're also getting functional annotations. So one of the benefits of using the protein pipeline is that a lot of these proteins have known functions. And when we get matches to those, we can assign these functional annotations to the different genes along the read as well. So you're getting a taxonomic profile as well as a functional profile simultaneously. So now I want to kind of discuss that filtering step in a little bit more detail and kind of highlight one of the improvements I've been working on for this pipeline. So recently I spent a lot of time adjusting the threshold filtering that's happening in Megan to obtain high precision and improved recall. And so I did this using the mock community data sets as part of the preprint that I just mentioned earlier. And so I calibrated these parameters using the mock community data sets, but below in this table, I'm actually showing the effects that this is having on an empirical sample. So this is the results for a human gut microbiome. And I'm showing these different abundance levels, so from high to ultra low, and then I'm reporting the number of species detected at each of these levels across the different parameter settings. And so the default setting, which I was initially using, ended up missing all species at low abundance and only partially detected the species present at medium abundance. And so this was not ideal. When I eliminated the filtering completely, I was able to recover all the species that should have been in the low and medium abundance levels. But in addition to that, I started picking up a lot of additional species at super low and ultra low abundance. And although this might seem like a good thing, in the mock communities, this was almost all noise and false positives. And so there was very little information here, and it was having a very negative impact on precision scores. And so I incrementally adjusted this parameter until I hit what I think is an optimal setting. And so here, we're detecting all of the species at low and medium abundance with high accuracy, and we're eliminating a lot of the noise that we're seeing at this ultra low level. So this optimized value is what we're using for filtering in the pipeline. But in addition to that, we also output the unfiltered version, right? In case that you're interested in seeing all the matches that were made to the reads, right? So you can choose between the two or use both or compare and contrast. That's really up to you, and it kind of depends on the use case that you have in mind, but both are available. So now I want to show an example of some of the results you can obtain using this pipeline. So here's an analysis that I did on several pooled human gut microbiome samples that were produced by the BioCollective. And these are, again, publicly available on NCBI. These are also mentioned in Jiawen's talk. So this was a really neat analysis because it contained 2 omnivore diet and 2 vegan diet samples, which allowed for meaningful group comparisons. So I ran all 4 samples through the profiling workflow, and I looked at the sort of a species level and genus level comparison. And I used the filtered outputs for both of these. So with the species level analysis, I had confident detection of 196 species in total. 91 of those species were shared across the diets. And then there were 105 that were unique to one diet or the other. So 57 unique to vegan and 48 unique to omnivore diet. So we can see some partitioning of species. And below I'm showing a heat map of all of those species across the different samples, with dark colors representing higher abundance and lighter colors representing lower abundance. And you can see that there are very clear differences in the relative abundance profiles across the diet types. And even in cases where we have shared species across the diet types, the relative abundances tend to be different. So we can kind of get this information on two different levels. And the same results are apparent in the genus level analysis. So here there are 81 genera in total. We find quite a few in common, but also some genera are unique to each diet type. And again, if we look at the heat map, we can see that there are clear differences in the relative abundances of these genera across the diet types, which is pretty neat to see. Okay, and so the last thing I want to mention is just some of the effects of sequencing depth on taxonomic profiling. So for this, we actually sequenced a new sample that we're very excited about. So this is the new Zymobiomics fecal reference with True Matrix technology. This is another pooled human gut microbiome sample. It's very high complexity and it's extremely useful for benchmarking as well as providing a reference standard. So we sequenced this sample using 4 SMART cells. And then I took that larger data set and downsampled it incrementally all the way down to a 96-plex. So anywhere from 88 to 0.3 gigabases of data. And then I ran each data level through the taxonomic profiling workflow. And below I'm showing the results of the optimized filter as well as the unfiltered results. And so on top, I'm showing the total number of species detected at each data level. And then the colors represent the relative abundance level, so high, medium, and low, the number of species contained in each. And we'll start out by just talking about this optimized filter. So somewhat surprisingly, I actually found that the information from 4 cells down to a 48-plex is largely consistent. So not only are the same species being recovered, but their relative abundance profiles are nearly identical. And so we're getting the same kinds of information from 88 gigabases as we do with 0.5 gigabases at the 48-plex level, which is really cool to see. If we look at 72 and 96, we start to see some variation in those profiles. And so we kind of draw the limit here at 48-plex. If we look at the unfiltered version, we see a much more dramatic effect, 7,000 species with 4 cells down to 491. But if you look at where these species are distributed, nearly all of them are coming from this ultra low and super low abundance level. And again, this is the level that contains a lot of noise and a lot of false positives, and is typically the relative abundance level that is filtered out prior to analysis. So I just wanted to highlight this as we are picking up a lot of additional things, but most of it is primarily noise. So at this point, given this downsampling experiment, we're pretty confident that we can multiplex up to 48 samples. And the next step that we're doing now is following this up in the wet lab so that we can confirm that these results are robust. And so this is good news for multiplexing and cost effectiveness in planning your experiments. Okay, so now I want to shift and focus on metagenome assembly. And so the goal here is to assemble a genome for each microorganism present in a microbiome sample. And here we're almost exclusively talking about bacteria and archaea. There are quite a few challenges associated with metagenome assembly. It's a really difficult task. So first, there's multiple species present, which throws off a lot of the targeted sequencing algorithms. There are uneven and unknown coverages for each of those species. And finally, there are lots of repeat regions as well as intergenomic overlaps within and between species and strains. And this can cause a lot of tangles and graph networks and make for a difficult task. But just like targeted sequencing assembly, the good news is that HiFi sequencing can also overcome many challenges involved with metagenome assembly. And so here I'm just illustrating some differences between assembling with short reads and assembling with HiFi reads. So with short-read datasets, after assembly, we end up with lots of small to medium-sized contigs. It's exceedingly rare, if not impossible, to assemble a genome into one single circular contig. And so we use binning approaches to sort of make sense of all this information. And here we're attempting to identify contigs belonging to the same putative organism and grouping them together. And the end result is what I would describe as draft quality MAGs. So each MAG can contain dozens to hundreds of contigs, and the potential for misbinning multiple species together, potentially missing information from the genome. But in the end, what we're looking at is highly discontiguous and fragmented assemblies for these MAGs. When we look at a HiFi assembly, one of the first things I'd like to point out is that the length of the reads, the HiFi reads themselves, anywhere from 7 to 15 KB, that's often comparable to the size of the contigs that have been assembled with short reads. So we're starting assembly at a fundamentally different point. And when we assemble HiFi reads, what we find is that we can get single circular contigs for a genome. And we also end up with, for the same reasons, lots of longer linear contigs. And so HiFi assembly also benefits from the binning approach. But here, the MAGs, we're getting more of them, and we're getting higher quality, with some of them approaching reference quality. So the pipeline that I want to talk about today is sort of the step between contigs and MAGs, which is focused on binning, QC, and ID steps. So again, this pipeline is also available on GitHub in the same location as our other pipelines. The name of this is the HiFi MAG pipeline. And we start this process using HiFi reads in a set of assembly contigs. So assembly has already been finished at this point. And this means that you can use any assembly method that you want. We've heard all of the benefits of using HiFi ASM Meta, and that's a strong recommendation. But you can also use HiFi appropriate settings in programs like Metaphy or Canoe. The first step of the pipeline is to map the reads to those assembly contigs to get coverage scores. And then we perform a binning step. And here we're actually using a custom circular aware binning strategy, which I'll describe in more detail in just a moment. After we make all the different bins, we want to assess the quality of each one. For this, we're using checkm. And we do have some default criteria that we're using to define high quality MAGs, but of course these can be adjusted depending on what your use case is. For all of those bins that pass this filtering step, we consider them putative MAGs, and we want to assign them to some type of taxonomy. And so for this, we're using the Genome Taxonomy Database Toolkit. And this can identify them to potentially species level, genus level, or family level, or higher, depending on how close of a match there is in the reference database. So the output from this is a set of high quality MAGs and associated metadata. So I want to focus in on this binning step in a little more detail and talk about some recent improvements. So we recently discovered this issue in the binning step where circular contigs were sometimes being misbinned with shorter linear contigs. And this is something that Xiaowen alluded to in her talk as well. So for example, if we take our full contig set, run it through MetaBat2, we end up with some bins that look the way we would expect, so several linear contigs being grouped together. We also sometimes see a single circular contig by itself in a bin, but then we get these cases where single circular contigs are binned with these linear contigs. And this has a negative consequence. So this increases the number of copies of what should be single copy genes detected in the bin. That inflates a contamination score, and then those bins are filtered out. So we're losing these otherwise perfectly good single circular contigs from this final bin step. And so there is a relatively simple fix for this, which I've called circular aware binning. So we perform the same binning step as we would normally with all contigs and MetaBat2. But in addition to that, we're identifying all the circular contigs from the assembly and then putting each one of those in their own bins. We're then taking these two distinct bin sets, we're comparing them and merging them and dereplicating them using DAS tool. And then they go through the same checkm filtering steps as before. And this ends up fixing the issue where we're losing these single circular contigs. We now get them recovered in our final bin set. Now, this is a relatively simple procedure, but it has quite a profound effect on the outcome. And so in my testing, what I found is it results in an 8 to 28% increase in total MAGs, but more importantly, it results in a 28 to 73% increase in single contig mags. And so this is automated in our pipeline, but if you're using your own binning procedure, I would strongly encourage you to use something similar to this, because most of the binning algorithms out there were not designed to work with contig sets that contain complete genomes and fragmented genomes. So this is a long read assembly specific problem, and it's something to be aware of. But if you use our pipeline, it's automatically implemented and it works extremely well. Okay, so now that we've gone over the pipeline in detail, I want to show some example results here. So I took one of those biocollective pooled gut samples, and I ran an assembly on it with HiFi ASM Meta, and then I ran the assembly results through our evaluation pipeline. And so after this was complete, I found 75 high quality mags, and below I'm showing plots which illustrate some of the characteristics of these 75 mags. So in the figure on the left, I'm showing the percent completeness versus percent contamination of the mag. And each dot represents an individual mag, and they're color-coded based on the number of contigs that they contain. So the best part of this graph is the bottom right-hand corner, which is high completeness and low contamination. And really the perfect result is to see a yellow dot here, which represents a single contig. And when we take a close look at this region, we find that there are in fact 28 single contig mags in this zone, which represent truly exceptional quality. And again, these came straight from the assembly process. Anything that's not yellow, which contains more than one contig, was recovered using that binning procedure. And so you can see that we have quite a few of those that we obtained in that manner, which maximizes our information. The plot on the right is showing the relationship between genome size and average coverage. And so these mags range in size from about 1 megabase to 5 megabases, and average coverage ranges from about 8x to close to over 100x. And the last thing I want to point out here is that I recovered 75 mags, but I only identified 66 species. And I found that several species had 2 strains present that were assembled to near completion. And doing comparisons among those strains, I found that there were some interesting differences in length as well as structural variation. And so it is possible to assemble distinct strains using HiFi ASM Meta, and we would expect that in this pooled sample, which is going to be more high complexity and likely to contain multiple strains per species. And the last thing I want to talk about is the effects of sequencing depth on assembly. And so again, I used our new Biomics reference standard to evaluate this. And so I took the 4 SMRT Cell data set and downsampled it to an 8-plex. And then I looked at the effects of assembly and evaluation on mag recovery for each of the data levels. And we know that assembly is much more sensitive to sequencing depth, and there is a very predictable relationship between sequencing depth and the outcome. So in blue, I'm showing just total high quality mag recovery. So at 8-plex, we have 9, and then you can see that there's this log relationship where we see rapid gains in mag recovery up to a single SMRT Cell. And then we see more diminishing returns as we increase sequencing depth, but still we're getting additional mags. And these are almost certainly representing the lower abundance species, which we now have sufficient coverage to assemble. So there's a log relationship here, but interestingly, when we look at the recovery of single contig mags, it looks like there's actually a linear relationship. And so with 4 SMRT Cells, we have 200 mags, we're getting 72 of those as single contig. But what's kind of crazy is that even if we look at the 8-plex, which is a very small amount of data to assemble, we have 9 high quality mags and 2 of those are single contig. So we're still getting good results even at this 8-plex level. However, if you are interested in assembly, and that's the main goal, our recommendation, of course, is to start at 1 SMRT Cell because you can see the clear negative impact on mag recovery here. And then if you need additional sequencing for lower abundant species, you can always increase sequencing depth. Okay, so to sort of wrap up here, the continual development of long-read metagenomics tools and workflows has resulted in precision species profiling, increased mag recovery and quality, and now, because of a lot of these deep sequencing experiments, we have a much better understanding of data requirements for each kind of analysis, and we can provide much clearer recommendations on this. Again, I want to emphasize that all of our pipelines are publicly available on GitHub. These are under active development, and so feedback is, of course, always welcome. And lastly, we have several HiFi shotgun metagenomic datasets that are publicly available, deposited on NCBI. If you go to the GitHub repo with our tools in it, there's a link to all of our datasets. You can find lots of information about each one and links to download. This includes mock communities, reference standards, as well as empirical samples. That includes human, chicken, and sheep gut microbiome. And then finally, we're really excited about this new Zymobiomics standard that we've sequenced. We're continuing to do additional sequencing on it. We've got lots of analysis underway, so that data set is not available yet, but we're planning on releasing it sometime in early summer. So stay tuned for that. And the big takeaway that I really want to emphasize is that with HiFi metagenomics, we can sequence more samples, recover more species, and recover more MAGs than previously before. And so hopefully I've convinced you of this, and I would encourage you to download some data sets and try out these analyses for yourself. And that concludes the talk, and I just want to thank you again for your attention and time.
Great, thanks, Dan. Awesome work. So we are at time, but we're going to go ahead and stay on for a few more minutes just so that we can answer some of you all's questions. If you do need to hop off, feel free to do so, but we're going to stay on for another 10 or 15 minutes to answer questions. So first question for Xiaowen is, so I'm just going to bend 3 questions here in one because they're related. How is the binning filtration done? And do you have to check for contaminations before doing the binning? And is there a good program for doing MAG binning using HiFi reads?
So for binning, we basically just follow the default Metaphy route, with the exception of splitting out the long circular conics after the binning. There's no pre-check before the binning process, and there's no other checks after the binning except for the actual evaluation process.
Okay, and do you have a decent program for doing MAG binning, or maybe what Dan mentioned is a suitable answer in his presentation?
Oh yeah, I think for pipeline, Daniel's pipeline looks very promising, and for existing tools, we used Metagenomic 2. It was kind of better than the other binners we have tried. I think we tried DAS tools. We tried sorry, I forgot the other one. But overall, the binning process is going to create a lot of contaminated bins, which bins multiple conics from different haplotypes. So I think it'll be good if anyone can figure out a way to split those bins. Otherwise, the common binners are all looking very similar, and I would suggest you to try all the binners and select based on check-in or other evaluations.
Okay, great. So another question related to binning is, binning in general struggles with single sample data sets, not necessarily due to HiFi assembly. Do you see better results from data sets with multiple samples? Any comment on that?
For multiple samples based on the human polled data set, it depends on how closely related are the samples. If I think if they're very different, then co-assembly doesn't complicate the process. But by pulling all the 4 human samples, we see that if it's somewhat related, then the assembler might be struggling with some shared areas. So for assembly, I would suggest to try single sample first. And because the whole assembly is relatively fast, you might also want to try co-assembly anyway, just to see what comes out. Oh, and for the binner, I recall that there's a few new binners based on neural networks. They're interesting, and the result was slightly better than Metagenomic 2, so you might also want to try that.
Okay, great. Another one for you, Xiao, and then we'll move to Dan's stuff. Would you expect genomes from different strains of the same species to collapse during the assembly, or would they produce different assemblies?
That's a good question. It depends on the sequence divergence, I suppose. So for the long circular conics, most of the time they're much separated from the beginning. By beginning, I mean after the error correction and the construction of the assembly graph. You will see it's almost circular subgraphs in the raw unity graph, and the graph cleaning simply steps through those subgraphs. So if the two strings are too closely related and they're tangled together in the unity graph, I think it'll be difficult to figure them out later. I think it's mostly empirical results. I don't have a clear boundary for that, but most likely we can only separate the haplotypes that have at least 1% whole genome sequence divergence. Lower than that, it'll be tangled together or collapsed.
Okay, great. Another question, are you able to characterize the virome from within metagenomic samples? What sequencing depth would be required to do this, or is it limited by the library representation? Have you seen any viral sequences come out of your assemblies?
Yeah, so for virome and also plasmids, we try to evaluate them, but unlike the genome sequences, we have checkm. For virus, there's checkv, but overall the reference dataset for evaluation is not complete, and the outcome actually depends a lot on what parameters you choose for the evaluation programs. So we kind of got a bunch of viral looking conics, but we are not sure how many complete virus were like the quality of those conics are. If you have a select deck, there's actually a table in the extra deck if you're interested in that, but overall we're not sure about the virus and plasmids.
Gotcha. Okay. Dan, so well, I can probably answer this one too. For read-based profiling, have we done any comparison with that from short reads? So a quick answer is yes. There are actually two papers in the handout section that you can download that address that. So the Gehrig et al. 2022 and the Portik et al. 2022 actually have comparisons to short reads for profiling. Dan, do you have any other comments on that beyond what I said?
Yeah, with regard to the number of species detected using each technology, they were comparable after the filtering. So Lumina and PacBio both give the same results and usually very consistent species. With regard to precision and recall and things like that, we always find that the short reads perform worse than PacBio reads. The reason for this is the K-mer approaches are not as accurate. And even after heavy filtering, we can achieve the same precision and recall that we can get using HiFi reads directly from the pipeline that I described. So it's always significantly worse.
And then for functional profiling also, there's much more reads that are being annotated with PacBio HiFi than there are with short reads. So it's usually 80 to 90% are being annotated to functions, and that's because of the long read links. They can actually get annotated to multiple genes at a time, versus 30 to 40% being annotated with the short reads. Another question for you, Dan. Did you evaluate how well the PacBio read-based profiling pipeline was at estimating the abundances of members of the microbial community?
Yeah, and that's something that I didn't show in this talk, but that's in the preprint as well. And a lot of the methods gave very poor relative abundance estimates, but both BugSeq and the PacBio pipeline, in most cases, gave very accurate relative abundance estimates. So they passed the goodness of fit test.
Okay. Most of the results focused on prokaryotic communities. Yes, they did, because these are all human gut. For metagenomic communities which contain eukaryotes, how do you predict HiFi reads perform in these cases? I have an answer to that. So there's a couple of papers that have been put out. Most notably I can think of is the Priest et al. Paper from 2021 that came out last July from the Max Planck Institute, where they did shotgun metagenome assembly and profiling on marine water samples, and they also compared that to short read data as well. So that's one to check out for that, where they have eukaryotes as well. Do you have any other comment on that, Dan, beyond what I said?
Yeah, and looking at a giraffe gut microbiome, we actually picked up several species of plants, which is kind of cool.
Trying to make it through all these recent ones that came in right at the end. What tool do you use to detect circular contigs from raw assembled contigs?
Oh, I think it's labeled by the assemblers in the output by default. You'll see if there's a suffix, or it'll be labeled in the annotation file.
Okay. Here's a question about assembling genomes of insects that sometimes contain lots of contaminants or cobionts. How would you recommend assembling these in these cases? So when you have, cobionts.
If those are not very similar to what you're targeting, I think you can just assemble the whole thing, and the assembler should be able to assemble those contaminants into like separate conics. If they are very similar, I think you still need to assemble the whole thing instead of trying to fishing out the reads. I'm not sure, because so far the empirical data sets, they don't have heavy contaminations. So I guess it all depends on what you get out of the assembly.
Okay, great,
So
thanks.
I would say too that I do have a little bit of experience with this, where we were assembling an insect genome and we were looking for bacterial endosymbiont. And I first ran the taxonomic profiling pipeline on it to identify potentially what that organism is. And once I identified what it could be, I then looked at the contigs that were assembled and fished those out by mapping them to the reference genome for that organism. After I identified that several big contigs were made, but it wasn't assembled to completion, I then used the contigs and I mapped the reads to the contigs. I was able to identify all the reads and pull those out separately and do a separate assembly, and I was able to assemble that bacteria into a single circular chromosome. So it's kind of a multi-step process, but it ended up working really well for that use case. And so you could potentially do something comparable.
Great, thanks both. For metagenomic assembly, are closed genomes from more abundant organisms, or do you also recover single contig mags for those low abundance organisms?
In the ATCC MOG dataset, it's actually interesting to see one of the most prevalent species being broken into two linear conics, probably due to the extremely high coverage. And it happened for all three assemblers at that place. In real samples, I don't think there's too much high coverage issues in the libraries. So basically, the more prevalent species will get a better representation in general. I think in sheep, we get long circular conics down to the coverage of maybe tenfold or something. But lower than that, it'll be difficult to, at least for getting long circular conics. For linear conics, sometimes it can go lower than that.
Okay, do you have any comment on that, Dan, from your experiences?
Yeah, I agree. I do think that if the organism is present in higher abundance in the sample, it's more likely to be assembled into a single contig, but that's not always the case. It depends on kind of the size of the genome and the complexity of the genome too.
Okay. Dan, for you, you're getting lots of questions around your circular contigs. So can you go in a little bit more detail on how you identify circular contigs? Is this a reference method? It seems like a lot of circular contigs are dropped in your normal process. So what are you doing that's different? Why is it so important? And why does this step complicate normal assembly, basically?
Okay. Yeah, so the circularity is reported in the outputs of HiFi ASM Meta, Metafly, and Canoe. So the pipeline actually, you sort of set which assembly method you used, and then the pipeline will automatically detect that information in the contigs that you supply. We then pull out all those circular contigs, sort of create a separate file with those, and then we have our original file that contains all contigs. And we treat those as separate to create those different bin sets. What happens when you misbin a circular contig with a linear contig is CheckM is looking for single copy genes. When you have more than one copy of those single copy genes, that affects the contamination score. And so adding anything to a complete genome will inflate that contamination score. When that happens, it filters out based on our quality control criteria. And so you end up removing that even though you have a complete genome present in the bin. And so binning these circular contigs by themselves is a way to circumvent that issue and sort of guarantee that we're going to recover those after the binning step is over.
Great, thank you. All right, I think we're going to end there. So in closing, I'd like to thank Xiaowen and Dan for their time today and for sharing their expertise and experience. I wanted to briefly mention 3 things before you leave, though. So first, in the next few days, we'll send you an email with a link to the recording, should you want to go back and listen to any part of this again. Second, you can stay up to date on upcoming webinars and events by visiting and bookmarking pacb.com/events. And finally, immediately following this webinar, you'll receive a very brief questionnaire. Please do take the 2 minutes that's required to fill it out, as it helps us understand your needs and plan for these future webinars. So thanks again for joining us today. I hope you'll join us again in a future webinar. And with that, take care and have a great day. Thanks, everyone.